Hello, I'm Kathy Lavoie. I'm speaking to you today about using ultraviolet C radiation to possibly control white nose in caves. My co-authors include Anna Pittis and Christopher Giagos, both students at the University of New Mexico. Alice and Eric are our National Park Service partners and collaborators, and Diana Northup from the University of New Mexico. Now, you're all familiar with white nose syndrome in bats. This is caused by an invasive fungus from Europe that was first found in caves in New York in the winter of 2006-2007. And it's now known as Pseudogymnoascus destructans, or PD for obvious reasons. And it kills hibernating bats. And in this picture, you can see some of the lesions on the wings and nose and ears of the bats. And it kills them by rousing them from hibernation so they don't have enough fat reserves to survive the winter. They wake up in the spring and go out to feed, and there's no insects there yet. White nose first recognized in New York State, and then has steadily moved out from that location. And unfortunately, we had hoped that it would not spread south because it's a cold-loving fungus. It only affects hibernating bats. But it kept going, and we thought it might stop around Mississippi because there's not many bats in the Great Plains, and it has continued its westward and southward movement. So there's increasing need for finding a way of controlling this infection. Now, Palmer, in a paper in 2018, found that PD was extremely sensitive to ultraviolet C radiation. And that's because it's missing some of the enzymes that cells normally use to repair damage from ultraviolet radiation. And he described it as a potential Achilles heel that might be exploited for treatment of bats with white nose or caves in our case. Now, this is an example of the electromagnetic spectrum, and you can see in the middle here we have violet visible light with the familiar Roy G. Biv breakdown of colors. To the right, we have longer wavelengths of radiation that have less energy, including radio waves. And then to the left of violet, we have ultraviolet, X-rays and gamma rays, and these wavelengths are getting shorter with more energy. UVA, UVB, and UVC are the primary forms of ultraviolet light, and they all work to kill cells by damaging the DNA. And when the DNA is damaged, then the cells can't divide. UVA and UVB are the ones that reach the surface of the earth and cause sunburns. And UVC, which has the highest energy, is actually completely absorbed by the ozone layer and water in the atmosphere, so it never reaches the surface of the earth. Now, some of the questions that we're looking at. First, can UVC be used to reduce the load of PD in caves? And related to that, what are the effects of UVC on native cave microbial communities? Is there going to be collateral damage if we use UVC in caves? And related to that, what is the sensitivity of isolated cave microbes to UVC? Now, in the process of doing the study, we're looking at three caves in the National Park Service, Oregon caves, which is a marble cave, caves in lava beds, which are lava caves, and Mammoth Cave, which of course is a limestone cave. But as far as we know, the populations, the diversity of microbes in these three different types of caves has never been compared before. And we're also learning old lessons again. We've used ultraviolet C radiation to disinfect for over 150 years, but there are certain tricks to using it, including shadowing and shielding. Now, shadowing is exactly what you think. There's something in the way that can block the UV radiation, and it doesn't reach the surface of the area that is shadowed. And anything can block this. We're all familiar with tan lines, so we know that UV does not penetrate through clothing, through your car windshield, through the lids of petri dishes. It just is easily blocked. Something that most researchers are not familiar with is shielding. And this is a situation where one cell can protect another one. And for example, if we take a bacterial cell that's 1 micrometer thick, it absorbs about 30% of the radiation that gets to the cell below it, which absorbs 30% to the cell below it, and so on. So obviously here the dose is important. The higher concentration you start with, the deeper the effects are going to penetrate, but it can be mistaken for resistance. Now, I put this in for a couple of reasons, because you're not used to thinking of bacterial cells in groups. Over here, this is an illustration of bacteria on the head of a sewing needle, so a very, very sharp needle. And here you can see a lot of individual cells, and then over here you can see clumps of cells that could offer shielding. And by comparison, I give you this illustration that shows the diameter of a human hair, which is about 70 microns. And if bacteria are between 1 and 2 microns thick, that means that a colony, the thickness of a human hair, is going to have 35 to 70 cells. So there's potentially a lot of shielding going on there. So a couple of the questions that we had to answer, what's the optimum concentration of cells to use, and what concentration of UVC to use? So we actually have a several step process. We dilute the cells, we plate them, and then we expose them to UVC, incubate them, and then take a look at what happens. Now this is a picture of the experimental study put together by Anna, and we mark off our petri dishes with 4 quadrants for streaking. We use this nifty little self-designed device on a printer, and we inoculate the bacteria into these quadrants after making our dilutions, and then we expose them to UV light. Now in this illustration, the lids are on. You have to take the lids off before you expose them to UV. And then we immediately wrap the plates in aluminum foil to prevent the light activation of repair enzymes. Now, this is an example of a dilution series. So we have test tubes that are each representing a 1 to 100 dilution. So we've plated them in triplicate on a plate of low nutrient agar. And this is the original, or this is our second tube, our third tube, and our fourth tube. And what we can see here is that we have optimum single layers of cells in the 10 to the minus 7th dilution. So this represents a dilution of 10 million from the original colony. Now, next, this is an example of a slow grower that's very sensitive from Oregon caves. And this is our control plate. So this is the original 1 to 100 dilution. And then after a 3-second exposure, a 6-second exposure, and a 12-second exposure to ultraviolet light in a biological safety cabinet. And what you can see is that there's no growth after 12, very limited growth after 6, and a huge reduction in growth after only 3 seconds. So we characterize this particular isolate as very sensitive. And we came up with criteria for sensitivity, mostly related to a 50% reduction in growth. If there's more than a 50% reduction after 3 seconds, we say it's very sensitive. If there's more than a 50% reduction after 6 seconds, we say it's sensitive. If there is more than a 50% reduction after 12 seconds, it's resistant. And if there's less than a 50% reduction after 12 seconds, we characterize it as very resistant. So here's some data, and you can see that this bottom line here, again from Oregon caves, there's about a 90–95% reduction after 3 seconds. So this one is characterized as very sensitive. This next one has a 50% reduction after 6 seconds, so it's characterized as resistant. And then these other two have more than, then this one has more than a 50% reduction after 12 seconds, so it's very resistant. And this is a summary of some of the data from ORCA, the number of very sensitive, resistant, and very resistant isolates. And what's interesting here is that the majority of the microbes are either resistant or very resistant to UV, which is pretty amazing considering they've been in caves with no exposure to ultraviolet radiation. We're in progress of doing this analysis on maca and lava beds, and that should be interesting. Now, I also said that one of the things we're looking at is comparing the microbial diversity in these 3 different types of caves. Lava beds is still in progress, but here we have results from ORCA and maca. And this represents a compilation of the 100 most unique species from each of the caves out of the thousands of isolates that we have. The ones in yellow are repeated on both lists. The column to the right here represents the number of isolates in that top 100. And the thing that immediately strikes me is that, of course, there's a lot of overlap, but there's also a lot of non-overlap. And in maca, everything is identified at least to genus, but at ORCA, we have several groups of unclassified, unidentified bacteria. So there's some unknown diversity there. So it's going to be interesting to see what we get out of lava beds. And Christopher Giagos is the student who's organizing this particular aspect of the study. Now here's some preliminary results of a study of the microbial community, and this was done at lava beds. And what's important to look at is this group of points down here. In blue, we have the diversity from our untreated sample, and then in red, this is the diversity that remains 1 hour after exposure to ultraviolet C radiation. And the important thing here is that there's no difference, no statistical difference, and none that we can see in this presentation of the data. And we actually attribute that to shielding, that in nature there is a thick enough layer of cells to protect the underlying cells. So our preliminary conclusions, UVC cannot be used to eliminate PD in caves. The surfaces are too rough, there's shielding, there's going to be shadowing effects, but it can reduce the numbers of PD on surfaces. So this is very similar to the current vaccination push to reduce the incidence of COVID-19. We're not going to get rid of COVID-19, but if enough people are vaccinated, we can reduce the incidence. If we can reduce PD on surfaces in caves, that will help to reduce the infection in the bats. And we also know that the native cave microbes show a range of resistance to UVC. So we think that collateral damage is probably going to be minor. And of course, fits in with the National Park Service tenet of do no harm. Work in progress and planned. We're going to continue with testing for UV sensitivity from the Park Service caves and then compare the diversity. We do plan on doing some more field work in the spring. We want to test the effects of UVC on cave communities at MACA, and then field test reduction from UVC on PD from MACA. So funding is provided through the National Park Service, and we have lots of people to thank with field work, and we will have many more to come. And are there any questions? I've put my email address up there. If you'd like to know more, feel free to contact me. Thank you.